Welcome to today's lesson on non-Mendelian inheritance. Gregor Mendel is famous for his experiments on pea plants, and he looked at 7 different characteristics: flower color, seed shape, pod color, pod shape, flower position, and plant height. As it turns out, all 7 of these characteristics followed a simple dominance inheritance pattern. In each case, there was a dominant allele, and it fully matched the expression of the recessive allele. For example, Mendel looked at plants that had purple flowers and white flowers, and he found that purple flower color was dominant, where white flower color was recessive. In real life, not all genes follow this inheritance pattern. In fact, other patterns are more common, and we're going to take a look at some other inheritance patterns that don't follow non-Mendelian patterns. They don't follow simple dominance. There is incomplete dominance, codominance, polygenic inheritance, and pleiotropy. The first is incomplete dominance. In incomplete dominance, one allele is not fully dominant over the other. The heterozygote is a blend of the 2 homozygous phenotypes. So if you look at this image here, there's a red flower and a white flower, and when they are cross-pollinated with each other, they produce offspring that are pink. So neither the red nor the white dominates. The offspring is a blend of the 2 parents, because if you mix the color red and white, you are going to end up with pink. The same can be said about these snapdragons. You have red, you have white, you have red, and you have the blended pink color. Here is a sample problem. When a white flower and a red flower are cross-pollinated, all of the offspring are pink. That is your clue that this is going to be an incomplete dominance problem because the offspring is a blend of the 2 parents. Cross 2 pink flowers and give the phenotype ratio. When we do problems with incomplete dominance, we're going to use a different notation. This isn't the only way to do this problem, but it is a way that works well. So the red allele we're going to indicate with a capital R. The white allele will indicate with a capital W. So the red parent is going to have 2 capital R's. The white parent is going to have 2 capital W's. And the offspring, who's heterozygous, is going to have 1 red allele R and 1 white allele, so 1 W. When we do our sample problem, we know we're starting off with 2 pink flowers, because that's what the problem told us to. And we already know their genotypes. They are each RW. When we put them into a Punnett square, we put the parents on the top and on the left-hand side. The first box gets RR, the second box gets RW, the third is RW, and the fourth is WW. Our first flower is going to be red, the second pink, the third pink, and the fourth is going to be white. The phenotype ratio is 1 red flower to 2 pink flowers to 1 white flower. Codominance is the second type of non-Mendelian inheritance pattern that we're going to look at. This is a pattern in which 2 alleles are fully expressed in heterozygotes. Neither allele is dominant or recessive. It's like they're both dominant at the same time. That's why we call them codominant. They're both going to be expressed. The heterozygote displays both alleles. So for example, this chicken is called an erminet chicken, and it has both black and white coloration. Its parents, one parent was white and one parent was black. And rather than it being a gray color, which would be an incomplete dominance, or either the white or the black dominating, both the black and white are expressed. Here's another sample showing you codominance. These are the parents, and the offspring ends up expressing both the red and the white coloration. Often with codominance, there's spots or there's some kind of spotty coloration. So let's do a sample problem with codominance. Black and white colored chicken feathers are codominant. What is the probability that a white bird and an erminet bird will produce white colored offspring? We're going to set up a similar notation to our incomplete dominance problem. We're going to use 2 different letters to represent our 2 different alleles. For the black parent, we're going to use capital B, capital B. For the white parent, we're going to use capital W, and for the erminet offspring, who are heterozygous, we will use a BW. In this problem, we're crossing a white bird with an erminet bird. The white parent has capital W, and the erminet has BW. Putting them into a Punnett square, the first box is going to have BW, as is the second. The third box, WW, and the fourth, WW. The first offspring is erminet. The second is erminet. The third is white, and the fourth is white. The phenotype ratio of black to white to erminette is 0 because there's 0 black-colored birds, 2 erminettes, and 2 white. So there's a 50% chance that the offspring will be white and a 50% chance that the offspring will be erminette. Human blood types are a special type of problem that involves codominance. They actually involve a multiple allele system. In this case, there is a gene for which there are 3 or more alleles that persist in the population at relatively high frequency. For human blood types, there are actually 3 alleles. The first allele we're going to indicate with a capital I, and it's going to have a superscript of an A. And this allele means that a person has A antigen in his or her blood. An A antigen is a substance, it's actually a carbohydrate that's inside of the blood of a person who has type A, for example. The second type of allele is I, so capital I with a capital B, and this allele has B antigen. This person has a carbohydrate in their blood called B antigen. The third version is a lowercase I, and this person or this allele has no antigen at all. So IA and IB are actually codominant, and the lowercase I allele is recessive. So you have 2 codominant alleles and you have 1 recessive allele. When we look at actual blood types and how they're formed, human blood types come in A, B, AB, and O. To get type A blood, there's 2 ways. You could have 2 of those capital A alleles, or you could have a capital A allele and a lowercase I. To get type B, it's similar. You need either 2 capital B alleles or you need a capital B and a lowercase I allele. To get type AB, there's only one way to do that. You need a capital A and you need a capital B. This person will have both A and B antigen in his or her blood. And then type O individuals have 2 lowercase i's. They carry no antigens in their blood whatsoever. Now, blood types, there's special cases, special blood types, and type AB is called the universal acceptor. A person who has type AB can get blood from anyone because he or she is not going to recognize any type of transfusion as being foreign. They will be able to accept blood from anyone. Type O is considered the universal donor, and the reason why is because type O doesn't have any type of antigens on it, so nobody is going to have an immune reaction to type O blood. They can just accept it without having a type of reaction reacting to that transfusion. So type O is called the universal donor. Now, type O can't get blood from any of these other blood groups because a person with type O will be sensitive to any type of antigen, whether that's A or B. So a person with type O can only get blood from somebody who is type O. So let's do a sample problem. What blood type will the child of a woman with type AB and a man with type O have? We have our mother, and she is AB, and we have our father, who is type O. We have to now write the alleles for the mother, and the alleles for the mother are capital I with a superscript A and capital I with a superscript B. I'm just going to call these capital A and capital B so I don't get tangled with the phrasing. And the father has 2 lowercase i's because that's the only way to get blood type O. When we put them into a simple Punnett square, we put the father's on the top and we put the mother's genotype on the left-hand side. Remember that this IA is really just 1 allele. This isn't 2 alleles. We're just using the A as a superscript. In the first box, we end up with capital AI. In the second box, same thing. In the third box, we have capital B lowercase I, and we have capital B lowercase I in the fourth box. Our first person is going to have blood type A. The second offspring, blood type A. The third possibility, blood type B. And the fourth is blood type B. So 50% of these offspring will have type A blood and 50% chance that they will have type B blood. This couple cannot produce offspring with type O blood, and they cannot produce offspring with type AB blood. Only individuals with type A or type B. Polygenic inheritance is the third non-Mendelian inheritance pattern we're going to look at. So a polygenic trait is a trait that's influenced by multiple genes. Usually, you can tell a trait is polygenic because there's a huge amount of variation in those traits. So one example is human skin color. Human skin color is determined by not just one simple gene, but by many genes. So there's a huge variation in human skin color, slight differences between people because this is a polygenic trait. Other polygenic traits include height, weight, eye color, and intelligence. These are all very complicated to determine their pattern because more than one gene is at play. So it's very difficult to predict the outcome of a cross with polygenic traits. The opposite of a polygenic trait is pleiotropy. This is a type of inheritance in which there's one gene that influences multiple traits. So just one gene, but it has a huge effect because it affects many different traits. If there's a mutation in this gene, that's really bad because that one mutation can affect many traits at once. An example of this is sickle cell disease. In this case, there's a mutation on the HBB gene. And multiple effects of this include, so first of all, the normal blood cells are mutated and they get this sickle cell shape. And this one gene causes a huge amount of damage because it causes an increased risk of blood clots, pain, organ damage, loss of vision, shortened lifespan, among other things. So that's pleiotropy, where you have a single gene, but it influences multiple traits. So I really hope that was helpful. We're going to spend a lot of time practicing these types of problems and we're going to get good at them. So thank you so much for listening.